Welcome to the unit on cellular communication. In this section, we will learn how individual cells are able to talk to each other over both long and short distances. The key topics include a general overview of cell signaling mechanisms, followed by a detailed look at cell signals and their receptors. Let's start with an overview. All cells, from prokaryotic bacteria to eukaryotic cells of multicellular organisms, are able to communicate. Cell communication involves four essential elements. The first is a signaling cell. This is the cell that initiates the communication. The second are signaling molecules, which are released by the signaling cell. You can think of the signaling molecules as the voice of the cell. The signaling molecule is then recognized by receptor proteins found on the surface of the responding cell. The receptor proteins are essential because they function as the ears of the responding cell. After the signal binds the receptor, it will cause a change, a response, in the responding cell. The response by the responding cell occurs in four steps. The first step is initiated by the binding of the signaling molecule to the receptor. This binding causes receptor activation. Once activated, the receptor transmits a message to the cell through the cytoplasm in a process called signal transduction. Signal transduction involves multiple proteins where the first activated protein goes off and activates a second protein, which then activates a third protein, and so on. This message can remain in the cytosol or go to the nucleus. The third step is the cellular response. The response will depend on what the signal-receptor pair was and what type of cell it is that is responding. Cellular responses can include activation of enzymes, changes in gene expression, production of additional signals, and can cause the cell to divide or change shape. The last step is then termination of the signal so that new signals can be received. An excellent example of communication between single-cellular prokaryotic bacteria is called quorum sensing. Quorum sensing is a way for bacterial cells to measure the number of cells in their population and cause a cellular response only when cell numbers are at sufficient levels. In this example, the bacterium Streptococcus pneumoniae constantly produces a signaling peptide which can bind to the receptor on the surface of cells. When the peptide binds, the bacterial response is to take up DNA from its environment to incorporate into its genome. When there are only a few cells in the environment, like there are here, there is a low concentration of the signaling peptide, and it diffuses away into the environment instead of binding to the receptor. However, if there is a high density of bacteria present, the peptide is made at high levels, and the result is a cellular response which causes the uptake of environmental DNA into the cell. In multicellular organisms, the distance between communicating cells varies depending on the cells that are communicating. There are four types of communication that occur in multicellular organisms. They are endocrine, paracrine, autocrine, and contact-dependent signaling. Signaling over great distances is called endocrine signaling. In endocrine signaling, the signaling molecule travels through the bloodstream using the circulatory system to move from the signaling cell to the receptor cell. Good examples of endocrine signaling molecules are estrogen and testosterone, which must travel from the signaling cells in the ovaries or testes through the bloodstream to target cells in various tissues. Paracrine signaling involves two cells that are close to each other, so the signaling molecule only needs to travel a short distance in order to bind to the neighboring cell's receptor. In paracrine signaling, the signal is usually a small, soluble molecule such as growth factors, which stimulate receptor cells to grow and divide. Autocrine signaling occurs when the signaling cell and the responding cell are the same. You can think of autocrine signaling as the cell talking to itself. Autocrine, as well as paracrine signaling, are both important during embryonic development. The last type of cell signaling does not involve a signaling molecule that is secreted from the signaling cell. Instead, it occurs because of direct contact between the signaling cell and the receptor cell. Contact-dependent signaling is essential for development of the central nervous system.